Hello, good morning, dear students. So today we are moving on to another topic, which is mutation. Mutation. Okay, mutation and the different types of mutation. We will see the different types of disorders due to mutations. Okay, so our topic is mutations. Like recombination, mutation is also another phenomenon that causes genetic variations. Mutation is also forms a base for genetic variations. There are favorable mutations are there, as well as unfavorable mutations are there. Genetic variations. Okay, mutations may lead to genetic variations. You might have studied in your 10th class about favorable mutations and unfavorable mutations. Favorable mutations, wait, 1 second. Yes. Favorable mutations leads to useful evolution, maybe for useful evolution, whereas unfavorable mutations may lead to diseases. Unfavorable mutations may lead to diseases. So now we will study about mutations in human beings. Okay. Diseases. Unfavorable mutations may lead to diseases. And here, what is a mutation? What do you mean by mutation? What is mutation? Mutation is a sudden genetic change, or it is a sudden heritable change, occurs in the genotype or genetic constitution of an organism, which is known as mutation. It occurs suddenly. That is why mutation can be defined as a sudden inheritable changes. It can be inherited also. Okay, genetic changes, sudden changes or heritable changes, sudden heritable changes occurs occurs in the genotype, that is in the genetic constitution of an organism. Occurs in the genotype of an organism, which is known as mutation. It is a sudden heritable change. It can be transferable to one generation to another generation, or maybe there is 50% possibility for that. In some type of mutations there is only 20% possibility, or in some other type of mutation there is 90% possibility for inheritance. Okay, so mutations are sudden heritable changes that occurs in the genotype of an organism. That means it is a change which occurs in the entire DNA. It changes, it alters the pattern of DNA. Okay, it makes changes in genes. Mutations are sudden heritable changes that occurs in genes. Okay, and that is alterations in DNA. It forms alterations in DNA. It is a sudden change occurs in the genotype of an organism, and if the genotype of the organism changes, that may lead to the change in the phenotype of the organism also. If the genotype changes, the phenotype also get changed. Okay. So that is mutation, and mutation is the chief cause. It is the main cause for variations, genetic variations. It is the main cause for genetic variations. So it is a phenomenon. Mutation is a phenomenon that causes alterations in DNA. It causes alterations in DNA sequences, and consequently it results in change in the genotype as well as the phenotype of an organism. Let me scroll the screen. Yes. So we can say mutations like this. It is a permanent change in the DNA molecule of an organism. Mutation can be considered as a permanent change. It may be a permanent change occurs in the DNA of an organism. DNA is nucleic acid. It is deoxyribonucleic acid. That is the genetic material in most of the organisms. In all the higher organisms, DNA act as a genetic material, whereas in viruses, RNA is a genetic material. So mutation causes a permanent change or alterations in DNA, in the structure of DNA, as well as in the function of DNA. When the structure of the DNA changes, that may cause changes in the functions or activities of the DNA. And the mutations occur very randomly. There is no particular chromosome to get mutated. It occurs randomly. Random changes. Mutations occurs randomly. It is random changes occurs in the chromosomes or genes, and it may be spontaneous, that is by nature, natural. It may be spontaneous, sudden, spontaneous, natural, or sudden. Spontaneous, or this mutations can be induced. It can be induced. That means the mutations can be also a consequence of certain agents. That means mutagens. The mutation causing agents are known as mutagens. Mutation causing agents are known as mutagens, and mutagens can be of different types. Physical mutagens are there, chemical mutagens are there, and that we will study that later. Okay, so here it is a permanent change occurs in the DNA, as well as mutation is a random. It is randomly occurring changes, or it may be spontaneous, or mutations may be induced. It can be, mutations can be created by some agents. That may be a chemical agent, or physical agents like UV radiations or X-rays, like that. And most of the mutations are heritable changes. Almost all the type of mutations except a few, okay, mutations are heritable changes. They can be inherited from one generation to another generation. So these are the peculiarities of mutation. Mutation means it is a sudden heritable change that occurs in a genome. It is a sudden heritable changes occurs in the genotype of an organism, and it may alter the structure as well as functions of the genetic material, that is DNA, in higher organisms. So the peculiarities of mutations are, mutations may be they are permanent changes that occurs in the DNA, and it is random. Random changes occurs, and maybe spontaneous, or sometimes the mutations are induced. That means it is caused by some agents known as mutagens, and these are heritable changes. Mutations can be inherited. If there occurs any mutation in one generation, that can be inherited to the next generation or to the next generation, like that. It can be carried on to generations after generations. And there are two types of mutations. Mutations can be classified into two types. Mutations are of two types. They are gene mutations and chromosomal mutations or chromosomal aberrations. Gene mutations or chromosomal mutations. Gene mutations or chromosomal mutations, or we can say that it is chromosomal aberrations. So, chromosomal mutations or chromosomal aberrations, that is damage or destruction, disarrangement of the chromosomes. Types of mutations, there are two types of mutation. One is gene mutation, another one is chromosomal mutations. So let us see what is gene mutation and what is the difference between gene mutations and chromosomal mutations. Gene mutations and chromosomal aberrations are of two types of mutations that we have to study, that it is very important. Gene mutations are also known as point mutations. Gene mutations. First one is gene mutation. It is also known as point mutation. Gene mutation or point mutation. What is that? These are the changes occurs in an individual gene. Gene mutation or point mutation are the changes it occurs in a single gene. Changes occurs in an individual gene, or it is the changes that occurs in a single base pair of DNA. In a single gene. It is a change which occurs in a single DNA. It is known as gene mutation, or it may be occurs in a single base pair. Changes occurs in a single base pair of a DNA. In a single base pair of a DNA. You know the structure of DNA that you have studied in your +1 in the chapter biomolecules. DNA consists of nitrogen base pairs, no? Consist of nitrogen base pairs. What are the nitrogen bases which are present in DNA? DNA, adenine, thymine, guanine and cytosine, and these always occurs in pairs. Adenine will combine with the thymine through a double hydrogen bond, and guanine may combine with the cytosine through a triple hydrogen bond. This is a base pairs present in a DNA. So if there occurs any change, any sudden change occurs in this base pair, that can be lead to some changes in the functionality of the DNA. That means adenine will always combine with a thymine and guanine will always combine with a cytosine. That means purines will always combine with a pyrimidine and pyrimidines will always combine with a purine. That is according to a scientist, Erwin Chargaff, which is known as Erwin Chargaff principle. So if there is any change occurs here, that means if the adenine combines with the cytosine, this is not possible. It is not at all possible because of the size of these nitrogen bases. And guanine can never be combined with thymine. This is also not possible. Or this thymine can never be combined with the cytosine, or guanine can never be combined with the adenine. All these are impossible combination. Only adenine can combine with thymine as well as guanine can combine with cytosine. So that is a basic rule. Always a purine will combines with a pyrimidine and pyrimidine will combine with a purine. Here adenine and guanine. Adenine and guanine are purines and thymine and cytosine are pyrimidines. So that is a basic rule in the formation of pairing of nitrogen bases in DNA, and if it pairs with any other molecule or any other nitrogen base, that may lead to mutation. That is maybe because of mutation. It may be because of mutation that may lead to dangerous effects later. Okay. So, and now we will see that the gene mutation or point mutation may occur due to any structural change in the DNA molecule at a single locus. Okay, so gene mutations or point mutations that may occur due to any structural change. It occurs due to any structural changes, that may be due to any structural changes in the DNA molecule, in the DNA at a single locus. In a DNA molecule at a single point or position, at a single locus. Locus means position or point. At a single locus. Locus means location, that means position or point. Position or point. That is why gene mutation is also known as point mutation. Gene mutation is also known as point mutation because it is a structural changes that occurs in the DNA at a single point or at a single locus, and they alter the message conveyed by a gene. Suppose this is a chromosome, this is a chromosome, and the centromere of the chromosome is here, and these are the chromatids, the arms of the chromosome like this, and our DNA or gene is located in the chromosome like this, and each gene conveys a message. Each gene conveying, it is conveying a message. That means depends on each gene or DNA forms all the characteristic features of human beings. So every set of genes, all these are genes present in a chromosome, and there each gene convey a message. Suppose this is a message to convey the what to say, hair color or hair pattern or height or eye color, and maybe this gene may convey the message for height, the character of height, or maybe this gene may control the features of eye color. So each and every characters in human beings, not only in human beings, in every organisms, it is controlled by different sets of genes which are present in the chromosome. And you just suppose if there occurs any structural changes, any alteration in any segment of these genes or in any segment of this chromosome, that may lead to mutation. That is, if it occurs in a single base pair of a gene, or in a point or in a single point, that may known as point mutation. It is known as the point mutation. Point mutations are they are limited to a single nucleotide. That means it is limited. Point mutations are limited to a single nucleotide. Point mutations are limited to a single nucleotide. I hope you know what is a nucleotide. What is a nucleotide? Nucleotide, that is a pentose sugar. Nucleotide means it consists of a pentose sugar plus a nitrogen base plus a phosphate group. This is a nucleotide, and here the pentose sugar plus nitrogen base, that is a nucleoside that you have studied in your +1 in the chapter biomolecules, isn't it? And when a phosphate group combines with a nucleoside, that forms a nucleotide, and many nucleotides join together to form a polynucleotide. And the polynucleotides, the polymers of polynucleotides, the polymers of nucleotides or the polymers of polynucleotides, they are DNA, nucleic acids, DNA and RNA. These are the nucleic acids. DNA and RNA are the nucleic acids. If you forgot those portions, please go through your +1 topics. Under the chapter biomolecules, it is there. You have studied that. Nucleotides and DNA as well as RNA, everything is there. So if there occurs any change in a single nucleotide, if there causes any change, any alternation in a single nucleotide, that may lead to mutation, and sometimes it may be harmful, but in most cases the point mutations are less harmful because it occurs in a single nucleotide. If it is occurs in the entire set of the chromosome or in entire genome sequence, that may be a very deleterious or harmful effect. That may lead to, it may cause a deleterious effect. And we know that gene mutations are sudden, they are very distinct changes occurs in the DNA, and that can be detected by, the gene mutations can be detected by visible changes in the phenotype of an organism. It can be detected by, the gene mutations can be detected by visible changes. Visible changes occurs in the phenotype of the organism. Phenotype means it is the observable features, observable expressions made by a gene. For example, height, we can differentiate the different contrasting traits of height into tall as well as dwarf. You know that. So that is a phenotype. And how the phenotype of an organism changes, because if there occurs any change in the genotype of the organism, that may lead to, definitely it may lead to a change in the phenotype of an organism. And so mutations can be detected by visible changes. If there occurs, if there is any change happens in the phenotype of the organism, that changes may be visible. All these changes may be visible. So the mutations can be detected by visible changes that occurs in the phenotype of an organism. Okay, that is, it is a change which occurs in the nucleotide sequence of the DNA molecule at a particular location or locus, and hence the whole message, what is conveyed, what has to be conveyed by the genotype, it will get derailed or it will change. The genotype change, that may changes the messages, changes the messages. What type of messages? Messages which are located, which are positioned in mRNA molecule, that we will study later. Don't get confused with mRNA here. Okay, so from the mRNA only proteins are forming. Proteins, all these proteins, whatever proteins are forming in our body, that regulates all the features, that controls or that regulates all the features in our body. And if there is any abnormal proteins are forming because of the alterations of the genotype, that may lead to, sometimes it may be harmful or sometimes it may be useful. So if it is abnormal proteins, definitely that may be harmful. If the genotype is changing, that may lead to the formation of abnormal proteins in our body, abnormal proteins, and these abnormal proteins may cause diseases or disorders that may be deleterious or harmful. Okay, my dear students, it is a very simple topic. If you study genuinely, you can learn it within no time. Okay, and the point mutations sometimes can be dominant or recessive or lethal or harmless. Point mutations can be, point mutations sometimes they may be dominant mutations, sometimes the point mutations are recessive. Some point mutations are lethal. Lethal means it is fatal. Fatal means it may lead to death. Fatal, that means it may lead to death. And some point mutations are harmless. Some are harmful, but some point mutations are harmless. And some point mutations are confined to X chromosomes, or it is restricted to X chromosomes, and such type of point mutations are known as X-linked mutations, X-linked mutations, X-linked mutations. And some type of point mutations are restricted to certain autosomes, autosomes, they are known as autosomal mutations. So sex-linked mutations, that means it may affect the sex chromosomes or allosomes, sex chromosomes or allosomes. allosomes, sex chromosomes or allosomes, and autosomes are not the sex chromosomes. They are the autosomes are located in the normal body cells. They are not present in the sex chromosomes. Sorry, sex cells. Okay. So 22 pairs of autosomes and one pair of sex chromosome, allosome, is present in human beings. And example, the very common example for point mutation is that you have to study in this chapter. The very common example is sickle cell anemia, that we will study. Sickle cell anemia. Sickle cell anemia. It is a disorder that affect RBCs. Sickle cell anemia is an example for gene mutation, or one famous, very common example for point mutation is sickle cell anemia. That means it is a disorder that affects the RBCs of human beings. And the gene mutations can be due to, there are many causes for gene mutations. The gene mutations can be due to duplication. Gene mutations may be duplication, that we will see in our video, duplication, and it may be due to insertion, duplication, insertion of a segment of a chromosome or gene, duplication of a segment of gene, or insertion of an additional segment of gene, or deletion if any chromosomal segment or if any gene is deleted, that may lead to a mutation which is known as deletion, or sometimes it may be substitution. Some segments may substitute some another segment. It may be due to substitution of bases, as well as there is the deletion and the insertion may lead to somewhat harmful type of mutation which is known as frame shift mutation. All these types of mutations we can watch in our video, frame shift mutation. So different types of mutations are there: duplication, insertion, deletion, substitution, and a frame shift mutation. All these are various types of mutations that occurs in human beings. Not only in human beings, in other animals also mutations may occur. Okay, so the mutagens, what are causing all these types of mutations? Mutagens are the agents that causes the mutation. Okay, so there are different types of mutagens. Physical mutagens are there, and chemical mutagens are there. We will study that. Physical mutagens and chemical mutagens are there. Physical and chemical mutagens are there. Mutagens are the agents, the substances that cause mutations are known as mutagens. Mutagens means it is the agents that cause mutations are known as mutagens. And here the chemical mutagens or the physical mutagens. First we will see what are the physical mutagens. Physical mutagens are X-rays, UV rays, all the radiations, X-rays, ultraviolet rays, UV rays, as well as gamma rays, and some cosmic radiations also, that may lead to some type of skin cancers, skin cancers, and different types of cancers are there. Cancers are actually because of mutations. The very harmful effect of mutation is a dreadful disease, cancer. Okay, so the cosmic radiations may also, these are some physical mutagens. All different types of radiations may lead to mutations. And chemical mutagens means any chemicals. For example, the chemicals which we are using in cosmetics, food industry, Food industry and detergent industry, detergents, whatever chemicals, manure, fertilizers, as well as some medicines, preservatives, that comes under food industry only. So all these types of chemicals are mutation causing agents. Mustard gas is there. And if we look carefully onto the packets of cosmetics or any other food items or fertilizers, whatever chemical, on the packet of that, suppose this is a tube of any cosmetic or anything, suppose if there is amase test positive, if it is written like this, amase test positive, that means this is a mutation causing substance, or it is a cancer causing substance, carcinogen. Carcinogen means cancer causing substance. So if it is written like this, amase test positive, that means that substance or any substance which is present in that particular cosmetic or that particular packet is a carcinogen, which is a cancer causing agent. That may cause mutations. So we are not aware of that. We doesn't care that. We are using different types of chemicals, or we are having different types of food and all. So sometimes it may lead to mutations. So that is about gene mutation. And now we can see what is chromosomal mutation. Chromosomal mutation. Chromosomal mutations, that is a mutations or the changes occurs in the structure and number of chromosomes. Chromosomal mutation, or we can say that it is chromosomal aberrations. Chromosomal mutations or chromosomal aberrations are changes. These are the changes occurs in the number. Changes occurs in the number and structure. Number and structure of chromosomes. Changes occurs in the number and structure of what? Number and structure of chromosomes. Changes which occurs in number and structure of chromosome. What do you mean by the changes occurs in number of a chromosome? That means normally human beings consist of 23 pairs of chromosome. 23 pairs of chromosome. Suppose if there occurs 24 pairs of chromosome, that may lead to mutation. That is a mutation. That is because of mutation. 24 pairs of chromosome. If it is 22 pairs of chromosome, that may also a condition which is known as mutation. And if the structure of, if there occurs any change in the structure of a chromosome, suppose this is a structure of a normal chromosome, and if there occurs any structural abnormality, suppose this chromosome is converting into a form like this. So one arm of the chromosome, a portion, suppose here there occurs a cut of this arm of the chromosome, and that arm, that chromatid may become the half of the size. So if this part of the chromosome is cutting and injured, or it is removed, that particular area of the chromosome is removed, this much segment of DNA is getting deleted from that part of the chromosome, and the remaining is only this much. Okay, so that is the structural changes which occurs in a chromosome. So see here the shape of this chromatid, this normal chromatid and the mutated chromatid. So this much segment of DNA is getting deleted from that. It is getting altered. So that occurs that is a change which occurs in the chromosome. And suppose if there is two arms, normally two chromatids are present like this. And suppose let me erase one chromatid from here. If this entire set of chromatid is deleted, that entire set of gene is deleted from there, no? Here there is no chromatid, that is deleted. So this is a structural abnormality, structural change that occurs in the chromosome, as well as if there forms any change in the number of normal chromosome number, if there occurs any change in the normal chromosome number, that may also because of mutations, or we can say that these are chromosomal mutations or chromosomal aberrations. And these chromosomal mutations or chromosomal aberrations can be detected under the microscope, but gene mutations cannot be detected by microscope. Here the chromosomal mutations can be detected by microscopic examination. It can be diagnosed under microscope. That means the chromosomal mutations means it may affect the genes or several genes. The chromosomal mutations may affect different parts, different segments of a gene, or it may affect many genes that may lead to very harmful effects. So chromosomal mutations are always causes many harmful effects. Chromosomal mutations may lead to very deleterious or harmful effects, diseases, very dangerous diseases. And these chromosomal mutations may be due to, here also this may be due to deletion. Chromosomal mutations may also occur due to deletion, insertion. That means each and every chromosomes or every genes which are present in the chromosome convey a specific message, and if there occurs any change, any addition or deletion or duplication of any segment of DNA or gene, that may change the message, that may alter the message, what should actually be transcribed. Okay, so it will alter the entire messages. As a result, abnormal proteins will be forming in our body, leading to harmful effects or diseases. So this may be also due to insertion, deletion, translocation. All these are the possible causes of the chromosomal mutations. And chromosomal mutations may also because of the change in number of chromosome that I have explained you now. Change in number of chromosomes, as well as because of the changes which occurs in the structure of chromosome. If the changes occurs in the number of chromosomes, there are different types of chromosomal number changes can occur. There are maybe because of addition or loss, addition or deletion, and these changes which occurs in number of chromosomes are of different types: aneuploidy and polyploidy, aneuploidy, polyploidy, and euploidy. There are different types of changes which occurs in the number of chromosomes. They are known as ploidy. Ploidy means changes. Changes which occurs in a chromosome. It is known as ploidy. Ploidy means it is a changes that occurs in a chromosome. So aneuploidy means it is a change in chromosome number which is not the exact multiple of the basic haploid number, because there is no change in the haploid number of the chromosome. But polyploidy means there forms a multiple changes occurs. There forms a very multiple set of changes occurs in the chromosome number, that is polyploidy, or it is also known as euploidy. Polyploidy can also be termed as euploidy, polyploidy or euploidy. Okay. And because of the changes of the chromosomal number, mutations are of different types. If there occurs any change in the number of the chromosomes, that may lead to conditions like trisomy, and monosomy, trisomy, monosomy, and nullisomy. Trisomy, monosomy, and nullisomy. And here trisomy means the 23 pairs of chromosome is a normal set of chromosome, and here that is a haploid condition, 2n number of chromosome, that is a normal. But in trisomy there is one additional chromosome will be there, that is 2n plus 1. This possess one chromosome extra, one extra chromosome. This one is an extra chromosome, one extra chromosome. And in monosomy means from the normal set of chromosome, one chromosome is getting deleted. That means 2n, that is a normal, minus 1, 2n minus 1. So 23 minus 1 may be 22. Here 23 plus 1, 24. So here there occurs one chromosome less. One chromosome is less here in monosomy. And in nullisomy means in a chromosome pair, chromosomes or genes or alleles may always occur in pairs. It may always occur in pairs like this. Chromosomes will always occur in pairs. Okay. So here in trisomy, this is a pair of chromosome. This is a pair of chromosome. And suppose if there comes one additional set of chromosome, that is trisomy. This is trisomy. And monosomy means additional set of chromosome is trisomy. Monosomy means this is normal. Chromosomes will occur like this. Normally it occurs in pairs. And here 2n minus 1. If suppose from here one is deleting or one is moving away, one is moving, so that may a single chromosome will remain there. That is monosomy. And in nullisomy, it is a chromosome pair out of 23 pairs of chromosome. Out of the 23 pairs of chromosome, if a pair is losing, that means nullisomy. That means 2n minus 2. 2n minus 2. Lost the both chromosome pair. That is nullisomy. Which is nullisomy. Okay, so the mutations which occurs in autosomes or normal body cells, general body cells, it is known as somatic mutations. Somatic mutations. Somatic mutation. One more type, that is somatic mutations. If it is somatic mutation, that means if the mutation occurs in general, my pen is very slow, general body cells, that is somatic mutation, general body cells of an individual. These are not transmissible or not inherited. Not inherited. These are not inherited and hence less harmful. And it does not play that much important role in evolution, somatic mutation. Whereas gene mutation occurs in the germ cells or gametes, sex chromosomes. They are inherited and these are harmful. They can pass from one generation to another generation and may be harmful, and they play an important role in evolution. So this is the difference between somatic mutations. Somatic mutations are the mutations that occurs in general body cells, and they are not inherited. They are not that harmful, and they are not the basis of evolution. Whereas gene mutations are mutations which occurs, or changes that occurs in the germ cells or gametes. Because these are the changes occurs in gametes, they are inherited and they may lead to evolution. They are the basis of evolution, and sometimes they may be harmful too. And spontaneous mutations are the mutations which occur by nature or naturally, randomly and spontaneously. They occur in a very slow rate, whereas induced mutations are the mutations that occurs because of certain agents which are known as mutagens, and they occur in a very high rate. Spontaneous mutations and induced mutations. Induced mutations are always caused by mutagens. Induced mutations. So this is all about the introduction, or this is all about mutation. Now you can watch a video for this. I will show you two videos are there actually. Mutations.
Mutation. Objective: to understand various types of mutation that occurs in the chromosomes. The complex human body is made up of millions of cells. Within each cell, we see a large prominent nucleus. Inside the nucleus of a dividing cell, we find 23 pairs of chromosomes. Each chromosome contains a large number of genes. Sometimes a segment of a chromosome comprising one or more genes gets deleted due to a mutation-causing agent. Thus, one arm of the chromosome is devoid of a gene, while the other arm possesses the normal copy of the gene. This type of mutation, which leads to the formation of an altered chromosome, is called deletion. In another type of mutation, a portion of the chromatid breaks at 2 points, excluding the centromere. This fragment rotates by 180 degrees and gets reincorporated. Thus, the order of the genes is rearranged in the mutant chromosome. This type of mutation is called paracentric inversion. But in another type of inversion, the breakage occurs in both the arms of the chromosome, and the broken fragment includes the centromere. This segment then rotates by 180 degrees and gets reinserted. This type of inversion, which involves centromere, is known as pericentric inversion. Sometimes a segment of one chromosome breaks and relocates on a non-homologous chromosome. This phenomenon can be described as non-reciprocal translocation. But in the reciprocal type of translocation, the two non-homologous chromosomes exchange a segment. Thus, the genes get altered in both the chromosomes. From a quick recap, we can understand that mutation can be defined as a rare, spontaneous, discontinuous, inheritable variation in the number or structure of the genetic material, chromosome, of a cell. There are 5 types of mutations, which include deletion, paracentric inversion, pericentric inversion, reciprocal translocation, and non-reciprocal translocation. Mutation. Object.
One more video.
Gene mutation. This topic explains the concept, types and effects of mutation. In our body, there are many eukaryotic cells. These cells are in a state of continuous division. Now, when we move inside a cell which is about to divide, we can see the nucleus and other cell organelles. Inside the nucleus, we can see some chromosomes. These chromosomes contain genes which are depicted in the form of bands. These genes code for different proteins. Any alteration in the genes is called mutation. Here, in the chromosomes, we find different base sequences that form various genes. To enhance our understanding, we use a sentence. The fat cat ate the wee rat as a sample gene. Point mutation. A point mutation is a simple change in one base of the gene sequence. This is equivalent to changing one letter in a sentence, such as this example, where we change the letter C in cat to an H. After point mutation, it changes to the fat hat ate the wee rat. Frame shift mutation. In a frame shift mutation, one or more bases are inserted or deleted, which is equivalent to adding or removing a letter in one sentence. This type of mutation can make the DNA meaningless. An example of a frame shift mutation using a sample sentence is when the T from the cat is removed. But when we keep the original letter spacing, after a frame shift mutation, the sentence is modified into the fat CAA TET HEWEERAT. Deletion. Mutations that result in missing gene are called deletions. These can be small, such as the removal of just one word, or longer deletions that affect the large number of genes of the chromosome. Deletions can also cause frame shift mutations. In this example, the deletion eliminates the word cat. Deletion. The fat ate the wee rat. Insertion. Mutations that result in the addition of an extra gene are called insertions. Insertions can also cause frame shift mutations, and it results in a non-functional protein. After insertion, it becomes the fat XLW cat ate the wee rat. Sex chromosomal aneuploidy. The 23rd pair of chromosomes in females is XX. while in males it is XY. Any increase or decrease in this number results in disorders. The Turner syndrome. It is the sex chromosome.
Yes. So the rest of the portion is the chromosomal disorders, various types of disorders, human genetic disorders, that is based on mutation. Mutations may lead to some disorders, genetic disorders. So we'll study about that. And human genetic disorders can be classified into Mendelian disorders and chromosomal disorders. So I'll write it here. Mutations were the last class. So now we are moving on to human genetic disorders. Human genetic disorders. Actually, dear children, I am not turning on my camera uh, because some issues are popping. Hmm? Hence, I don't want to turn on my camera. You just listen the voice. That is enough. Hmm? Actually, I wanted to take class on the board, but some problems are there. That is why I am not turning on the camera to take the classes on board, on blackboard, the wall board. Not taking classes on the wall board because I don't want to turn on my camera. Yes. So here there are a number of disorders caused by genetic abnormalities. We know that. Many varieties, many types of disorders are there. Many genetic disorders are there. Genetic disorders. You might have heard about Down syndrome, mental retardation, so and so. So all these are some genetic disorders. There are there are number of genetic disorders or the disorders which are caused by genetic abnormalities. Genetic abnormalities. Disorders which are caused by genetic abnormalities. And among those disorders, some of these may be due to the incompatibility of the gene. It is because of the inefficiency or the incompatibility of the gene products. That means proteins. Gene products are actually proteins. It all the genetic abnormalities may be they are the reasons of some incompatibility of gene products. Gene products are proteins. Gene products, they are the proteins. So in our body, if abnormal proteins are forming, that may lead to genetic abnormalities. If abnormal proteins are forming, if the proteins formed in our body are abnormal or indeficient proteins, abnormal proteins, that may lead to, it may lead to genetic abnormalities. Genetic abnormalities. That may lead to genetic abnormalities. Okay. Yes, and these genetic abnormalities sometimes passed on to the next generation, successive generations also. The some genetic abnormalities may be very defective or dominant or recessive. Genetic abnormalities may be dominant or recessive. Sometimes it may be very harmful, or sometimes it may be minor. Harmful means sometimes that may lead to death, or it is lethal. Death causing means it is lethal. 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 Lethal means it may cause death. Okay, so the genetic disorders may be grouped into two categories. Genetic disorders can be grouped into two categories. They are Mendelian disorders and chromosomal disorders. Genetic disorders. Genetic disorders are classified into two types. They are the Mendelian disorders and the chromosomal disorders. Mendelian disorders means these are the genetic disorders that follow the laws of inheritance. Mendelian disorders. It follows the laws of inheritance, Mendel's laws of inheritance. So the Mendelian disorders and chromosomal disorders. The genetic disorders are classified into two types. One is Mendelian disorders, the other one is chromosomal disorders. Mendelian disorders are the genetic disorders which follow the principles of inheritance, which follow the laws of inheritance. Laws of inheritance. Laws of inheritance, it occurs in autosomal genes, autosomal cells, and it is determined by alternation or mutation in a single cell. These Mendelian disorders are caused by mutations occur in a single cell. Yes, my pen got stuck. Let it come, let it come. So the Mendelian disorders are determined by any mutations. It is determined by any mutations or alterations in a single gene. Alteration in a single gene. Whereas chromosomal disorders are due to changes that occurs in the chromosome number as well as structure of the chromosomes. Today first we will study about the chromosomal disorders. After then we will move on to genetic disorders. And let me first explain about a few points also about genetic disorders. Genetic disorders strictly follows the laws of inheritance, that is Mendel's laws of inheritance. It is happened by any mutation or alteration in a single cell. It can be transmitted from generation to generation, and it can be determined by, it can be traced by or diagnosed by pedigree analysis. Pedigree analysis. The Mendelian disorders can be diagnosed by, or the disorders can be traced by pedigree analysis. Pedigree analysis. And the Mendelian disorders may be due to the presence of recessive or a dominant gene on autosomes. It is due to the presence of recessive gene or dominant genes. Recessive or dominant genes which are linked on autosomes. Dominant genes present on autosomes, not the sex cells. Autosomes. Autosomes are somatic cells, they are the general body cells. Or sometimes it may be linked to sex chromosomes also. Okay, so the Mendelian disorders can be caused by recessive or dominant genes which are present on the autosomes or sex chromosomes or X chromosomes. Sex chromosomes as well as X chromosomes. X chromosomes. Okay, so that is about the Mendelian disorders. And today, now we are going to study about the chromosomal disorders. The second one, that is chromosomal disorders. Chromosomal disorders are, maybe it is due to the changes. These are due to changes in the number of chromosomes. Changes occurs in the number of chromosomes, or also by structural changes in number as well as structure of chromosome. That is chromosomal disorder. Changes in the number of chromosomes, that is, in human beings the normal number is 23 pairs of chromosomes are present in human beings. So if there occurs any change in the number of chromosomes, that may lead to chromosomal disorders. And if there occurs any change in the structure of chromosome, that may also lead to chromosomal disorders. Suppose this is a chromosome with a centromere, and here the structure of the chromosome changes. Means, suppose here you look into this picture, that is, I'll represent it in another color. Suppose this blue colored arm of the chromosome, blue colored arm, this is just for your reference only. This blue colored arm of the chromosome is this much length, isn't it? And if any mutation occurs and this arm of the chromosome, this chromatid of the chromosome is injured, suppose it got injured, and what happens? That arm is got injured, and if it is forming a structural change, let me take another color. Yes, all the other arms or chromatids are perfectly all right, there is no problem for other arms. But here in this diagram, my pen is not working properly, my dear students. That is why it is getting delayed. I need to take blue color to show the blue chromatid, blue arm of the chromosome. It is not coming. So suppose this blue, yeah, it is coming. This blue arm of the chromosome has got injured, and the length of the chromosome is reduced. See the difference between the two arms, two blue arms or chromatids of the chromosome. Here in this diagram, it is about this much. But when mutation occurs, it is only this much. The tip, the top portion is cut. It is injured. It is got injured, and it is eliminated. Suppose this is that injured part. Okay, so that is a sort of chromosomal disorder. This may lead to chromosomal disorder because this is a sort of structural change that occurs in the chromosome. So the chromosomal disorders are caused by additions of a pair of chromosome that may lead to change in the number of chromosome, or it may be due to structural changes of the chromosome. Herein the changes in number can be occurred by addition of extra pair of chromosome, or deletion of extra pair of chromosome, or inversion of the chromosome, or because of translocation of chromosome. All these things we were discussing in our last class, no? So now we will see the examples for the chromosomal disorders. Examples for chromosomal disorders. The chromosomal disorders are of autosomal disorders, and sex chromosomal disorders. It may also affect the autosomes as well as in sex cells, sex chromosomes. So first we will study about the autosomal disorders. Autosomal chromosomal disorders. Autosomal chromosomal disorders. some examples. You need to study an example for autosomal chromosomal disorder is Down syndrome. It is Down syndrome. It is also known as Mongolism or trisomy. Mongolism or trisomy. The cause of this autosomal disorder is, autosomal disorders are the disorders which affect the autosomes of the body, autosomal genes of the body, the genes which are present in autosomes of the body. So here in the Down syndrome, you know, it is a sort of mental retardation, no? Down syndrome, it was discovered by, the Down syndrome was discovered by a scientist, a physiologist, and a geneticist. His name is Langdon Down. It is there in your textbook. His name is Langdon Down. Down syndrome. Langdon Down. So the scientist who discovered Down syndrome is Langdon Down. And it is due to the presence of an extra chromosome on the chromosome number 21. Chromosome number 21 got an extra chromosome. That is why the chromosome number 21 has a two pair of chromosome like this. This is a normal condition, two pair of chromosome, but there forms an extra chromosome. So the chromosome number 21 got one additional chromosome. So altogether it is the chromosome number 21, 21st chromosome, 21st set of chromosome will be like this. There will be 3 chromosomes. So this is known as, this disease is also known as trisomy 21. Trisomy 21. Because there is an extra chromosome at the 21st position of the chromosome map of all the chromosomes. In human beings, you know, in human beings we have 23 pairs of chromosome. First one, second, third one, fourth one, fifth one, like that it is going. And this arrangement is known as karyotype. Likewise, 21st, 22nd, 23rd pair. And here in the 21st position, this is happening. That is, one extra chromosome occurs there. Hence, this condition is also known as trisomy 21. And these individuals have, because of the extra chromosome, such individuals possess 47 chromosomes altogether instead of 46 chromosomes. The normal chromosome number is 46 number of chromosomes or 23 pairs. 46 numbers, or it is 23 pairs of chromosomes. That is a normal number. Instead of that 26 number of chromosomes, in the individuals shows Down syndrome, they possess how many chromosomes? 47 chromosomes, because of the extra chromosome which is present at the 21st position. That is, the chromosome number 21 gain, or it got one extra chromosome. So this disorder can also be referred as trisomy 21. Trisomy 21. Yes, dear students. And why this happens? Why this is happening, Down syndrome? The Down syndrome is it is because of the unequal distribution of chromosomes during gametogenesis. It is because of the undistribution of chromosomes. This board is not, the trisomy 21 happens, it is because of the unequal, it is because of the unequal distribution of chromosomes, unequal distribution of chromosomes during gametogenesis, during egg formation or gametogenesis, specifically during egg formation, that is oogenesis, during egg formation or oogenesis. That is, during gametogenesis the chromosomes segregate or separates, meiotic division occurs, reduction division occurs. If you don't remember gametogenesis, please go through human reproduction chapter. Please go through the chapter, second chapter, that is human reproduction. There you can see gametogenesis and both spermatogenesis and oogenesis. There the meiosis also is there. Meiosis, that is a reduction division. There you can see that. Okay, so that segregation or separation does not occur in such disease. Hence, one extra chromosome is transferring from the parent generation to the offspring. And that non-segregation of chromosome is called non-disjunction. Non-disjunction of chromosome. That is the non-segregation or the chromosomes are not separating, or there is no reduction division. It is non-segregation or separation of chromosomes during gametogenesis. That is known as non-disjunction. It is known as non-disjunction. Normally, during meiosis, there is an equal distribution of chromosomes to each daughter cells. That everybody know. In normal meiosis, I will write. In normal meiosis, what happens? In normal meiosis, an equal distribution of chromosomes, equal distribution of chromosomes occurs because of the segregation or separation. But if there occurs any mutation, what happens? There won't be any segregation or separation of chromosomes during gametogenesis. That may lead to an extra number of chromosome in some positions of the chromosome, of the entire chromosomal pattern. That here in Down syndrome, in the case of Down syndrome, the extra chromosome occurs at the 21st position. 21st chromosome consists of an extra chromosome. That is a non-equal distribution of chromosome. That is, in trisomy, the normal separation of chromosomes fails. In trisomy, it is a condition where the normal segregation or separation of chromosomes fails. Fails to occur. Okay, the normal separation or segregation of chromosomes fails to occur, and here instead one pair of chromosomes remains together with an additional number of chromosomes. So that is trisomy 21. Trisomy 21, it is an autosomal genetic disorder. It is caused by an extra chromosome at the 21st position of the chromosome pattern, and the number of the chromosomes can be represented like this: 2n plus 1. 2n is a normal number plus 1, that is an addition or extra, that is 46 number of chromosome is a normal plus 1. So in individuals with a trisomy, how many number of chromosomes will be there altogether? 47. That is an abnormal condition. Now we can see the symptoms of Down syndrome. Symptoms of Down syndrome or this particular disorder. Severe physical and severe physical. There are many symptoms occurs in case of Down syndrome. Some symptoms are discussing here, that is severe physical and mental retardation, physical and psychomotor, severe physical or psychomotor, that is mentally retarded and mental retardation, mental retardation. That is the first and foremost symptom is a very severe physical imbalance or psychomotor imbalance as well as mental retardation. All these conditions are very severe, and certain abnormalities, certain number of abnormalities are also occur in the facial structure of such individual. Facial structure, that is the pattern or the face structure, facial structural changes. Some changes occurs in the facial structures, like very flattened nose, very flattened nose or nasal bridge, very flattened nasal bridge, and the wide opened mouth, habitually open mouth. Such individuals open their mouth during a long period of time, partially open mouth with a furrowed tongue. Partially, the mouth is partially opened with a furrowed tongue, and the very prominent head, protruding head, prominent head, prominent head, very prominent head or protruding head, and the lower lips are protruding, prominent or protruding head, and the lower lips are protruding, very prominent or protruding head and prominent head, and protruding lips, protruding or projected lips, protruding lower lips, the lower lips are protruding, and they consist of a furrowed, they have a furrowed tongue, furrowed tongue. That means we can see a sort of division or margin in the tongue. Suppose this is a tongue, here you can see at the tip of the tongue a furrow like structure like this. So a protruding and a furrowed tongue is present in such individuals, and some characteristic fold of skin at the corners of the eye. Folded skin, where? At the corners of eye, corners of eyes, both eyes, and they are very short structure, very short individuals, and with a very broad neck, with very broad neck. I think all the symptoms are there in your textbook also. And another symptom is very broad palm crease, very broad or widened palm, broad palm crease, very broad palm crease, with very short fingers, widened palm crease with very short fingers, and they may be associated with many heart diseases, by birth itself. Their heart will be very defective, that is congenital heart disease. Congenital means by birth, inborn heart diseases. So all these are some features of characteristic features or symptoms of Down syndrome, the individuals with the Down syndrome. And always slanting eyes, slanting eyes. So these are the examples. This is one example for autosomal genetic disorder, that is Down syndrome, and the symptoms of Down syndrome are these are the symptoms of Down syndrome. Okay, and now we will see sex chromosomal disorders. So this is autosomal disorder. Down syndrome is an autosomal disorder, and the genetic disorders are classified into two: autosomal disorders and sex chromosomal disorders. So Down syndrome is an example for autosomal disorder. Now we will see the sex chromosomal disorder. Sex chromosome disorder. Sex chromosome disorder. These disorders, the sex chromosome disorders, are occur due to abnormal number of sex chromosomes in the cell, because of the abnormal number of sex chromosomes. Abnormal number of sex chromosomes in the cell. It is because of the abnormal number of sex chromosomes, not autosomes, abnormal number of sex chromosomes in the cell. And this abnormal number of sex chromosome is also because of non-disjunction or non-separation or non-segregation nor non-disjunction of chromosomes during gametogenesis. It is also because of the non-disjunction or non-segregation of chromosomes during gametogenesis. And we all know that in human beings, the sex determinant is the Y chromosome. Y chromosome is a male determinant. The presence of Y chromosome gives rise to male individual in appearance. Okay, irrespective of the number of X chromosomes present. And some examples, and the absence of Y chromosome leads to a female child, female individual. It may give rise to a female individual. And we can see some examples for sex chromosomal disorders. That is, first one is Klinefelter's syndrome. Klinefelter's syndrome. Klinefelter's syndrome. Example for sex chromosomal disorder is Klinefelter's syndrome. Klinefelter's syndrome. Klinefelter's syndrome. It is due to the presence of an extra copy of X chromosome, because of an extra copy of X chromosome. Extra copy of an X chromosome. So the individual may consist of 47 number of chromosome, that is XXY, and this X is an extra copy of an X chromosome. So the altogether the number is 2n plus 1, that is 46 plus 1 is equal to 47. Here additional copy of an X chromosome. The normal, the individual, if it is a male, it is XY. Suppose one X chromosome is added, that is an addition. So XXY, that may forms an XXY. So this is Klinefelter syndrome. This may lead to a chromosomal disorder, a genetic disorder, that is Klinefelter syndrome. So the karyotype of the chromosome is XXY. XXY. The individual is a male and has overall masculine development will be there. It may be the individual is, the offspring is a male with masculine structure with masculine features. Masculine features will be there, but some features of feminine features may also will be there in such males. Okay, because of the additional X chromosome, additional or extra X chromosome. That is, the individual will be males, but they have a defective or underdeveloped testes. Underdeveloped testes. Underdeveloped testes and very little body hair. Very little hair on the body. Mentally retarded. Very little hair on body, very little hair on the body, and they may be mentally retarded. Very little hair on the body. Such individuals may be mentally retarded. Individuals may be mentally retarded, and such individuals may show a very peculiar feminine character, that is development of breasts or mammary glands. I am waiting for the red color to come on. So these are the symptoms of Klinefelter syndrome. Masculine features are there, but underdeveloped testes, mentally retarded, very little body hair, and development of breasts or mammary glands. And such condition is known as gynecomastia. Such males have mammary glands or breast like females. Development of breast. It is because of the additional X chromosome, additional number of an X chromosome. So the feminine characters, feminine features, it is development of breast, and that condition is known as gynecomastia. And such individuals will be sterile. That means they have no capacity to reproduce. They do not have reproduction capacity. Such individuals may be sterile. So that is Klinefelter syndrome. These individuals may be sterile. They do not have the reproduction capacity. So that is one sex chromosomal disorder. It happens in the sex cells, non-disjunction of the sex chromosomes. And the second example for sex chromosomal disorder is Turner syndrome. Second one is Turner syndrome. Let me scroll my board. It is bit slow because it is stuck. I don't know what happened with the whiteboard here in the computer. Wait for a second, dear students. Yes. So the second example for sex chromosomal disorder is Turner's syndrome. It is caused by the deletion of one extra chromosome due to the deficiency or lack, absence of one X chromosome, absence of an X chromosome, Turner syndrome. It is because of the absence of an X chromosome. Hence, the individual has only 45 chromosomes. And the karyotype can be represented as X0. It is because of the absence 2n minus 1, that is 46 minus 1 is equal to 45 number of chromosomes. 2n minus 1. Such individuals may be females, but they are sterile. They don't have the capacity to reproduce. They cannot reproduce. Reproduction capacity will not be there, and such individuals are sterile because they have underdeveloped ovaries. Underdeveloped ovaries, that means their ovaries are not developed perfectly. They have underdeveloped ovaries, hence it may interfere with their oogenesis. Ovum production may not be there. Underdeveloped ovaries. Ovaries are not well developed, or it is not a proper ovary. Insufficient ovum formation occurs there. There won't be any ovum formation or oogenesis. That is why such individuals will be sterile. They don't have the reproductive capacity, and they have very poorly developed breasts. Very poorly or underdeveloped breasts. Poorly developed mammary glands. Developed breasts or mammary glands. Ovaries are underdeveloped, breasts or mammary glands are also underdeveloped. And very short stature, very short individuals, very short stature, short stature means short structure, very short stature or height. With a webbed neck, that means two foldings will be there in their neck. Webbed neck or folded neck. Folded neck, webbed neck or folded neck. They cannot menstruate. Menses will not be there. They cannot menstruate or ovulate. Ovulation or menses, menstruation may not be there in such females. So that is Turner's syndrome. Short stature, very rudimentary or very underdeveloped or poorly developed ovaries, webbed neck, and they will be, such individuals are mentally retarded. Mental retardation. Mental retardation also there. Mental retardation. So this is Turner's syndrome. Turner's syndrome. So these are all the chromosomal disorders, and in our next class we will be studying about some Mendelian disorders. Here today we were discussed three disorders. One is Down syndrome, that is an autosomal disorder. It is a sort of chromosomal disorder because of the additional number of chromosomes. And the second one is sex chromosomal disorders. Under sex chromosomal disorders we discussed the two disorders. One is Klinefelter syndrome and another one is Turner syndrome. In Klinefelter syndrome also there occurs an additional number of X chromosome. So the individuals may be males, but they have feminine features. And the second one is Turner syndrome, that is because of the deletion or absence of one X chromosome. One X chromosome will be deficient or lack or absent. So the karyotype, the representation of the chromosome will be X0. Such individuals are females with one X chromosome with lack of one X chromosome. So X0 is the condition. So such females are sterile because of the underdeveloped ovaries, and they cannot menstruate or ovulate. Ovulation will not be there, and they have underdeveloped breast or mammary glands with short stature and webbed neck and mental retardation. So all these are the chromosomal disorders. Now I think I will show you one video here. Chromosomal abnormalities and chromosomal disorders. Let me check whether it is there. Yes, chromosomal disorders.
Meaning an extra complete set of chromosomes. Chromosomal disorders you have learnt in scientific mostly in plants. In this, the failure of cytokinesis after the telophase stage results in the daughter cells gaining an extra complete set of chromosomes. In this module, you have learnt, in scientific terminology, the presence of a defined set of chromosomes in each cell is called ploidy. Ploidy is of two types, aneuploidy and polyploidy. Aneuploidy occurs when the chromosomes fail to segregate, as a result of which the daughter cells lose or gain chromosomes. Aneuploidy is of two types. Hyperploidy, it is a decrease in the normal somatic chromosome number. Hyperploidy, it is an increase in the normal.
All this we have discussed in our last class also. Polyploidy, aneuploidy, etc.
Somatic chromosome number. Polyploidy is a condition that occurs mostly in plants. In this, the daughter cells gain an extra complete set of chromosomes. Mendelian disorder, sickle cell anemia.
So, Mendelian disorder we didn't study today, and we were discussing about chromosomal disorders now. Let me check whether it is there in this video. Otherwise, we will see it in our next class. Chromosomal disorders. Let me check whether it is
such as diabetes, thalassemia, etc., can all be inherited.
Yes, Down syndrome is there.
Down syndrome is a genetic disorder caused by the presence of an extra chromosome, resulting in mental retardation and malformation in newborns.
Dear students, here you can see the karyotype of the individual. Karyotype means it is the entire set of, it is a pictorial representation
At times,
of the
during the
entire
fusion
set
of
chromosome. Karyotype is a pictorial representation. It is just like a graph or map of the entire set of chromosomes in an individual cell itself.
ovum and sperm, the gametes accidentally contain an extra chromosome in the 21st pair of chromosome, since it does not divide during meiosis. This results in a cell containing 24 chromosomes, while the other has 22 chromosomes. This accidental event is called non-disjunction. Since the 21st chromosome is an added chromosome to the pair, Down syndrome is called as trisomy 21. When this defective chromosome is inherited by the baby, he has 47 chromosomes in all. The children with Down syndrome are mentally retarded, malformed, and also suffer from cardiac problems, respiratory infections, leukemia, and reduced lifespan. In this module, you have learnt the following. The processes of meiosis as well as fertilization result in varied combination of genes. Mutations, too, cause variation in genes and chromosomes. A change in the gene structure or a chromosome, or even the sequence within the chromosome of an organism, resulting in the creation of a new trait different from the parental type, is called mutation. Mutagens are substances or agents that cause mutation. These agents can be in physical or chemical form. Down syndrome is a genetic disorder caused by the presence of an extra chromosome, resulting in mental retardation and malformation in newborns. When the gametes accidentally contain an extra chromosome, as one pair does not divide during meiosis, and thus one cell contains 24 chromosomes and the other 22. This is known as non-disjunction. Since the 21st chromosome is an added chromosome to the pair, Down syndrome is called as trisomy 21. The children with Down syndrome are mentally retarded, malformed, and also suffer from cardiac problems, respiratory infections, leukemia, and reduced lifespan.
Nucleus chromosome. Genetic material called deoxyribonucleic acid. 1064 plants. If a karyotype shows an extra chromosome, while the D group has 3 pairs, even the E group has 3 pairs. If a karyotype shows an extra chromosome in the 21st pair, the condition is called trisomy. This disorder causes Down syndrome. Through the study of karyotype, we can study the various genetic diseases. Recap.
Yes. So, dear students, that is enough for today, and we will see the rest of the videos in our next class, as well as the rest of the portions by our next class. Okay, thank you.